I'm here today with Rosemary Sinclair Dokos and Lakmal Jayasinghe. Tell me, Rosemary, what is it that you do for the company?
Thanks, Zoe. So I'm our Chief Product and Marketing Officer. In that role, I lead our product teams, our marketing teams, as we build and develop the products that we deliver out to our users. I've been here for 10 years, since launching the MinION into the ravenous mapper community all the way back then, and have loved every single step of the journey, from those 500 early users to the multitude of users that we have today, the multitude of products and the multitude of applications and geographies that we serve. And it's been a truly humbling experience to be here.
Fantastic. Lakmal?
So I'm the Chief Scientific Officer of Oxford Nanopore. I've been here with the company for more than 18 years. My job is to make sure that the scientific vision and the scientific approaches of the company is fit for purpose. So from genomic, when we move from genomics to more multiomic approaches, so there's a long way to go. And my job is to make sure that we create these new opportunities with scientific development within the company.
Fantastic. And you are known, you're a very modest man, but you are known as one of the world's best nanopore scientists. So we should probably mention that about you. So, Rosemary, what is your kind of manifesto? What are the kind of things that we have to deliver in the coming years ahead?
Sure. I boil it down to three things. We have to build great products. We have a fantastic platform, really highly differentiated, delivers really rich information very, very quickly, is very affordable. It has to be paired with a fantastic experience. And so that's not just on the product side, that's on how you order, that's how you get supported. That whole piece needs really sort of bringing together and coalescing. We are not going to stop innovating. So we are absolutely going to deliver the best product, best performance, best value for our customers. So we will keep driving our accuracy, we will keep driving our outputs, we will keep driving the novel information that you can get out of the platform. We'll push the multiomic piece really, really hard. And then the final piece is sort of that building for impact, getting this technology out of the labs where it is today and into the field where it can have the biggest impact. So into the hospitals, into the diagnostic labs, into the doctors' offices, into the factories, into the dentists, where this is really going to truly change lives. And so those are really the sort of the three things: great products, best platform, and highest impact.
Fantastic. So, Rosemary, you've talked a lot about the fact that you're really passionate about customer experience. You want every person to move through the journey with us frictionless, and really, really happy. How do we meet that challenge that we have ahead of us?
It's a really big challenge, because we make our sequencers incredibly affordable, but they are still sequencers. They can produce incredibly powerful biological information. And so what we're working on doing is packaging up what currently happens in the industry, where people are investing in large instruments, and that comes with support, and that comes with engineering time and support staff, which we do have for our larger installs. But we're trying to package up that entire experience and serve it digitally, so that anyone who buys a MinION has a best-in-class digital experience and gets up and running quickly and is incredibly successful. And that is a big challenge. It's not an easy thing to do, but it's something that we're really passionate. We think we can do it.
And how does the customer voice come into that? How are we going about that challenge?
We listen to customer input from everywhere, not just sort of social media, but every single case that comes in. Every single customer visit, any sort of recommendations about how we can make products better, that all feeds in. So we're always listening.
So, Lakmal, you've been with Oxford Nanopore 18 years, and I'm sure there's another 18 in there with all the things that we have opportunities to develop on this platform. How do you think about the talent, the scientific minds that we need to gather to fulfill all of those opportunities?
Yeah, so when we started the company, everybody thought nanopore sequencing can never be done, but we proved them wrong, because we have a fantastic set of scientists coming from multiple disciplines. We have biologists, chemists, biochemists, molecular biologists, data scientists, software people, engineers. They all individually improve their pieces of science, and then they all need to come together. So the collaboration is very important. So we work with quite a lot of academic collaborators, but we also work with our industry partners, and also like over the years, as our name as Oxford Nanopore, as one of the smartest companies in the world grows up, it makes us easier to attract talent. So I actually led the internship program at Oxford Nanopore, and last year, for example, we had 24 positions to offer. And we had 1,400 applicants, and they are fantastically talented. And the pool that we have to choose was very difficult to select people because they were that good. And when I mentioned this to Hagan Bailey, the founder of the company, he said that is even getting into Oxford Nanopore internship program is even difficult than getting into Oxford to do a degree. So that's a testament to our R&D talent. They are fantastic, and I really enjoy working with them.
And how do you provide leadership to the team, because they're so diverse, all of these scientific skills? How do you unite them against these common goals that you have? What are the tricks that you use?
Yeah, so that's the challenge, and because we attract the top talent, and you can't treat them like technicians, they need the independence to develop science. So we give them the guidance in terms of like overall, this is the company objective and this is the group objectives. So each quarter we have quarterly objectives, and then I make sure that they are aligned with company objectives, and then each team leads make sure that the work is getting done, and then we discuss and we collaborate, and even though like very different disciplines, they all come together and make science work.
Fantastic. So Rosemary, your teams are both innovating, but also you're working across teams who are building products like Q-Line, destined for regulated environments to be used in the same way every single time. How do we think about culture and processes when we're juggling those two things?
Yeah, it's something actually that we had to really take head on, and I think for that reason we came up with those sort of release mechanisms, which are sort of the beta releases, the early access, the full released and Q-Line, so that for every single project that we're doing, we know what the route to market is, and we know if we're serving an innovation customer, the teams can serve that really quickly. If we're serving a regulated customer, it has to go through more and more development. We've brought in fantastic talent with regulatory experience, but also platform development experience. We've married that talent with people who've been at Nanopore for many years, for sort of 12, 15 years. And there's not a solid wall between the development groups and the regulated development groups. There's, those teams interact actually on a very regular basis, almost on a daily basis. They're interacting as the regulatory team are trying to understand how things work, how are things developed, and taking that through to a regulated pathway. So there's a lot of respect, and actually what we try and do is figure out what are there things that the regulatory team do that if we did more in the innovation side would actually accelerate innovation, and are there things that we would do in the innovation side that would accelerate the regulatory. So those teams actually work very, very well together.
So we're a growth company, we're an innovation-driven company, but we're also a mature company that has to be extremely responsible about how we direct our resources into that growth. So how do you make your resource choices in the scientific teams, Lakmal?
So as you said, Oxford Nanopore is a very innovative company, and innovation is at the heart of our research and development program. Even though we have a mature product, we still do not stop innovating. Even for DNA and RNA sequencing, we still do quite a lot of R&D work. And then going forward, like for other multiomics development, we still need to do quite a lot of work. So when we decide where to invest in terms of resources, it's really coming from the experience we had, everything we learned over the last 18 years, and also we are deciding how the applications or future changes For example, currently like machine learning is very important, and even in DNA sequencing, RNA sequencing, and going forward for all other omics applications, we need to have a lot of like machine learning involvement. So we recognize these unique features that we need to add to our pipeline, and then we invest in the right time and the right place. It is expensive, but we have to do it, and we do it well.
Excellent. Rosemary, a lot of investment choices to make in our teams. How do we think about it?
Yeah, so I think there are long-term investments that we're really behind, and those are things like our voltage sensors, things like our distributed sensors that will enable even more distributed sequencing, and those are sort of quite easy. That's a long-term vision of the business. The more immediate term, again, it's sort of that power of having sort of the marketing, the product teams working really closely together. Because this platform can do so many different applications, it's always a matter of prioritizing. What are the applications where the technology is most suited today, where there is a good market, where there's a good appetite for change, and really bring those teams together and create those end-to-end workflows that will empower our commercial group to deliver on the numbers that we need them to deliver for the growth of the company. But so yeah, the long-term strategic visions are really clear. We want to enable the sequencing of anything by anyone anywhere. Very clear. Short-term, we work a lot with our marketing groups and bring together our products and workflows team to make sure that we have the right products to make our commercial team successful and our customers successful too.
Fantastic. So you've just come back from Boston. We had the Nanopore community meeting. Hundreds of Nanopore users in the room sharing best practice. And you gave a wonderful update about what we've been focusing on this year and what we're delivering for that user base. So Lakmal, in your talk, you talked a lot about RNA. What did you talk about in your update, and why is RNA particularly exciting for you?
Because for a long time it was just the genomic era. People were just sequencing DNA, but then it became transcriptomics, which is RNA sequencing, became very important. Currently, like with the COVID developments, everybody knows how important mRNA vaccines are, and the biopharma industry at the moment trying to use our technology to quality control of vaccine batches. Currently, they have to use multitudes of different technologies, a lot of human resources, and it takes long time, weeks and months to quality control one batch of vaccines. With Oxford Nanopore sequencing technology, we are the only technology that allows you to sequence RNA in natural version directly without converting them to cDNA. So with our direct RNA sequencing technology, all that experiments can be replaced by just one single nanopore sequencing method. So that is very exciting, and we have worked so hard over the last year to improve our performance of direct RNA sequencing, and now it's in a place that all these biopharma companies can use to improve their quality control approaches.
So what's changed in the last year then?
So we improved our accuracy significantly, about like 4 to 5 Q points, and we improved our output. We improved for all these QC approaches. There are like certain elements of testing that they need, and we addressed each one of those one by one, and then we are now in a really good place to drive this biopharma QC.
Fantastic. Well, I think as an innovation and development engine, and indeed a production engine for the biopharma industry, it's really exciting work that you're doing, and very valuable to the industry as well. So Rosemary, in your talk, you talked a lot about how we improve the overall user experience as well as the performance of the technology, and of course you talked a lot about high throughput human genome sequencing. What were the highlights of your talk, do you think?
Yeah, that's right. So a lot of our customers and a lot of people who've heard of Oxford Nanopore hear a lot about the capabilities that the product has and how you can do many, many, many things with it. And what we've been really focused on with our groups over the last year in particular is how do we take powerful workflows, so biology that a lot of people want to look at, and how do you productize that? How do you make that simpler and more accessible to more people? And we've been launching many, many workflows this year, from our bacterial assembly workflow, Nomis, that went out earlier this year, to plasmid sequencing that's been out for a while. One of the things that we've worked on extensively with customers now is the ability to sequence lots of genomes. Of course, people have been sequencing human genomes since 2003. That took 10 years and hundreds of people, but now you can do a human genome in a lab, particularly thanks to the P2 product. But we have a growing appetite from users to sequence, to go from hundreds of genomes to thousands of genomes. And in a production environment. And so that means really sort of pushing up the number of genomes you can do, pushing up the robustness. And so our teams, our development and product teams worked really hard on creating a workflow, a sort of completely packaged workflow together with customers like Precise that we've been working with extensively this year, so that customers can sequence hundreds of genomes per week. They can reproducibly get the 30x coverage that they all want. And also because now we've implemented a barcoding solution, they can start pushing in terms of the numbers of genomes that you can run and start reducing the flow cells that they're running them on. So actually start getting a better and improved price per genome by running this high throughput barcoding application. The other really exciting thing that we've been doing is taking our machine learning prowess that we've developed over the years that have been really focused on base calling and starting to point some of that talent into things like assembly, sort of read polishing, and also variant detection, variant callers, and starting to improve the performance of analysis tools outside our direct sphere of influence. And that is so that we can bring all of those improvements back into these well-packaged workflows and deliver really what is an outstanding human genome at scale, and actually at any scale for any user. So we really focused on that in what feels like the second half of the year, but it was actually the summer because NCM was in September this year, and the teams have worked really, really hard on it.
Fantastic. And you've also talked about telomere to telomere, telomeres being the ends of the genome, so our ability to see everything, not just the 92% of the genome that could previously be seen in one go. Is everyone using telomere to telomere, or could everyone be using it in the future, or why is that important for us?
There's an enormous amount of appetite as the T2T consortium has grown. People are starting to understand that it is of high value to have a sort of more custom reference to each project. And so all of these large projects where people are sequencing thousands of genomes, they are all, the starting point normally is a reference. And what we're starting to see now is that all of these genome programs want a telomere to telomere reference that they generate so that they can then analyze the data against their own reference. And so the appetite is there. The excitement is great to see a lot of people talking about Nanopore only T2T, because that's made it far more accessible than the previous versions. And the fact that you get state-of-the-art assembly stats with just one platform. And that's been an enormous amount of collaboration from the chemistries that enabled it, that have come out from the research teams, all the way to the informatics pipelines that have come together, and the whole productization again of something that a few people could do before, but now we want many, many more people to do it. And so we're productizing all of these exciting innovations that come out.
It's really exciting to see more communities who have more diverse genomic data sets. We all know that traditional data sets were really focused on the sort of Caucasian groups, and to see more diverse communities driving their own reference genomes and their own genomics programs. And it's just great to be enabling that. So you must have had a favourite customer talk or a cluster of them in Boston. What was your highlights, Lakmal?
There was the really interesting talk from a 16-year-old student for the first time ever. I mean, we talk about sequencing being anyone, anywhere, by anything. And this kid gave a talk how he used a Min9 to analyze water samples, and he was remarkable. And to see something like that enable just a young student to go and do something that he wants with the technology that we develop, that's really exciting. So in the biopharma day, there were quite a lot of talks about sterility testing. So sterility testing is when you inject a biological sample, whether it is a vaccine or any other cell therapy drug, you need to make sure that it is very sterile, which means it does not contain any microorganisms like DNA or any virus, bacteria, or any other microorganisms. So currently in the industry, they need to use quite a lot of different techniques to validate that. But now there is a drive to use long-read nanopore sequencing to understand each biological sample better and actually what's in there. So that is quite exciting.
So Rosemary, there's a great cancer track at the Nanopore community meeting. What were the scientific highlights coming out of there?
That was a great track. There was interesting talks from brain tumor, intraoperative brain tumor surgery, into all things like single cell as well. What was really brilliant was to start seeing a sort of a shift in the cancer community from what is today. There's an enormous amount of cancer sequencing, but it's all sort of amplicon driven. It's all sort of yes no questions. And it's really interesting to see a subset of that community start to push the knowledge into this really challenging disease with the information richness that Nanopore provides.
So Rosemary, we all talk about accessibility. Was there anything of particular interest at the Nanopore community meeting that talked about the importance of accessibility in science?
I thought the first talk was actually a lot of fun. It was a professor who had a student who said that they wanted to use Nanopore, and the professor said, no, it's not going to work. It's not accurate enough. But said, I will give you the opportunity. This was a sort of a brand new postdoc. I'll give you the opportunity and the funding. Go and prove me wrong. And the student did prove him wrong and came out with phenomenal assemblies, incredible performance, and really changed this professor's mind to the point that he was the plenary speaker at a Nanopore conference just mere months after this event. And I think that really sort of speaks to the power of the technology. Of course, it's empowering a lot of people, a lot of sort of very, very large labs, but also that ability for a student to come in and say, I want to try something. The fact that it's affordable and accessible means that you can try it and create fantastic science out of it. And that was it really linked into the fun, the amazing 16-year-old that we had later on in the conference. So it was a really, really beautiful talk.
I think there wasn't a single person who came out of that conference who didn't mention Noah, the 16-year-old, and I think we were all slightly put to shame by his prowess as a student. So it's a perfect segue to talk about multiomics, Lakmal. I know this is one of your deep passions, the ability to see lots of different types of molecule and to analyse them. Is it as simple as swapping out the nanopore and swapping the enzyme, or is there something that a bit more complicated than that you and your scientific teams have to achieve?
Very good question. It is not as simple as that, but our platform is very modular, so we can actually take one pore out and put another pore in if we change the chemistry from genomics to transcriptomics to proteomics. It is simple as that in a way, but then you have to develop all the chemistry around it as well. But the cool feature of our platform is that we don't have to redesign the entire system because it is very modular. You can swap components very easily. So that's why we can actually push this multiomics approaches as quickly as possible. So we will be probably the only company that can do all genomics, transcriptomics, proteomics, metabolomics all in one platform. So that will be a very, very powerful position to be. So that's what I'm going to push in the years to come.
So obviously part of that is in the chemistry and the biology, as you say, but a lot of that is in the data analysis. And we're lucky enough to live in a world where AI and machine learning and an enormous computational power is being developed every day. How are we harnessing those kind of technologies, and how is your scientific team thinking about data analysis as one of our sort of tools in our toolkit?
Machine learning is actually becoming quite central to everything we do. Not only for engineering of proteins, and we use machine learning quite a lot to improve our pores and enzyme, and also, as you say, to analyze large data sets. So this week, for example, Nobel Prize went to protein designing using machine learning tools, which is quite exciting. So we work with some of these key groups, and we have our own machine learning team as well. So we use all that knowledge to improve our chemistry and to improve our data, improve our data analysis. And going forward, when we try to design chemistry for proteomics, metabolomics, like we will use every inch of machine learning available to us.
So we design in silico before we design in real life, is that correct?
We do, yes, correct.
Amazing. Thank you very much for speaking to me today. I'll let you get on with all the innovation that we have to do in your day ahead. Thank you very much. Bye.
Thank you.
Thanks, Zoe.